lucky today actually to have Susanne Schmid with us. Susanne is an assistant professor Department of a tenured position in the Department of Anatomy and Cell Biology, and also director of the neuroscience program as well. And as we've just been discussing, Susanne got her Ph.D. at the Eberhard Karls University in Tübingen, Germany, and her current interest is in cellular and molecular mechanisms underlying sensory filtering in mammals. And so Susanne and I have been working together with a graduate student on sensory processing and autism, and are there ways to actually measure that, and then how we can use that information to perhaps look at interventions and how things change over time biologically. So Susanne will talk to us about that today. Thank you. Thank you for coming.
Thank you. Okay, so as Rob said, my background is actually biology. So I study really neural function in the brain in rats. I was initially when I started working with my own group, I was interested in how synaptic plasticity, underlying learning, things like that. But through branches and things that happen in life, moving to a faculty of psychology in Toronto, and then medical faculty here at Western, the framing of my work shifted. So now it's all about sensory processing and sensory filtering in animal models for autism. And as Rob also said, we have a student at the moment who also works with children with autism and or ADHD. That's why I advanced this. Does this work?
Sarah is theoretically in charge of this.
Yes. Power switch? Yeah. There you go. Okay. Okay, so sensory disruption or impairments or changes in how sensory information is processed have become, with the DSM-5 now, one of the core symptoms in autism spectrum disorders. And if you look at the literature about sensory issues connected with autism spectrum disorders, there's often a report of both. Is there a laser pointer? Yeah.
I think in the government of Ontario we're not allowed to use it. I'll say that's the reason anyway. It doesn't work.
So we often read about hyper and hyposensitivity. So that seems to be a contradiction, but actually it's not. I think because for one, it's autism spectrum disorder. So there are, as probably you all know, very different manifestations of autism. And so some of them might show hyper and some of them might show hyposensitivity. But even if in the same child or in the same person, we often can see both hyper and hyposensitivity. And we work a lot in the auditory acoustic field. And there, and you will see that later as well, but we often observe that there's a hypersensitivity to not so loud, so to quiet and mid-range intensity stimulation, but then a hyposensitivity to the loud ones. And so the same person can have hypo and hypersensitivity. You also have to keep in mind that all sensory receptors tend to adapt upon constant or repetitive stimulation. And so sometimes, and might be, one example might be these weighted vests that some of these kids like, these kind of stimulations might also lead to receptor adaptation. So a strong stimulation might lead to receptor adaptation and actually work against a hypersensitivity. Although you would think they are hyposensitive because they want strong stimulation, they sometimes might use that to actually dampen their sensory stimulation by receptor desensitization or receptor adaptation. So this confusion about are they hyper or hypo is maybe caused by either of these three things, that they are different patient populations, but also depends on the stimulus intensity, and it depends on receptor adaptation or desensitization. In general, I think all senses can be affected. We hear about touch things, we hear about acoustic, visual. So the only thing I haven't heard much is about smell. Are they sensitive to smell?
I think some kids Well, I mean, it's all filtered through parents' information, so it's always hard to
Yeah.
know. But at times, parents will certainly say that kids can't, if they're in certain smells. Yeah, well, I mean, it's not,
It's secret, right?
Yeah.
Okay.
Okay, so all senses can obviously be affected, and the problem is exactly that often there's no systematic assessment of sensory issues. It's all, or most of it is parent questionnaires. So it's just reporting the reports of the parents that you can't really quantify that. Which is a pity because it could actually also be a part of the diagnostic marker. So if a kid is hypo or hypersensitive, and what kind of stimulation that could maybe help to differentiate the different subtypes within the autism spectrum. So my background again is biology. So I myself always have been interested in sensory processing and most importantly to sensory filtering. And if you look at these sensory issues that parents report or that you might observe, then we really have to break it down in 3 different domains. So one is the sensory processing itself. So the primary processing of, for instance, acoustic information on their way up to the cortical levels where there is a perception, there might be alteration in the sensory processing pathways themselves. But there also might be alterations in sensory filtering, and I will quickly explain what that is, or in reactivity. So maybe their sensory processing is fine, and their reactivity, their reactions to sensory stimulus is different. So and this is hard, of course, to tease apart, or we can't tease that apart with parent questionnaires. But there are ways to actually measure different things. Just quickly about sensory filtering. So this is really the core theme of my research group, is that idea that we have 5 senses and we have a lot of sensors inside of our body, and they basically bombard the brain constantly with sensory information. We cannot shut any of our senses off. We can close the eyes. It's debatable whether we probably still see something. So that huge amount of sensory information has to be filtered and reduced in order for the brain to be able to make sense of things. And there are different stages where it's filtered. There's receptor adaptation, as I mentioned. So constant or repetitive stimulation will lead to some of our sensory receptors to adapt. They become non-responsive, or they respond to a lower level. And then at the very other end of the sensory processing pathways, there's attention, where we can voluntarily focus our attention on something and try to ignore the rest. But a lot of sensory information is filtered in between. So these are, along the sensory processing pathways, they're built-in neural mechanisms that filter out and gate sensory information, so that only the important stuff will actually use our valuable resources in our brain and go up all the way to the cortex, where they then will be actually perceived. So a lot of sensory information get lost on your way, and this is what I'm looking at, which is my research topic always has been: how does the sensory processing, the sensory filtering in these lower areas, brain areas, happen? What are these built-in mechanisms? How are they disrupted in autism or in schizophrenia? And these built-in mechanisms are pre-attentive, so it doesn't require learning, and it does not require any kind of consciousness or perception or effort. It is inbuilt, and it happens way before any sensory information ever reaches the cortex and anything that conscious perception. So that happens. These mechanisms are built into all of your brains, into my brain, and they normally work, but they do not work in people with autism, schizophrenia, some other neurological disorders. So from now on, I want to focus on the acoustic sensory portion, because that is what we work mostly, but not entirely. We do a little visual as well, but we mostly focus on the acoustic part. Did I take my one?
Do you want to use your families?
Hmm?
Did you say you have one?
No, I have one as well. Maybe I should use mine.
But it's not in the budget. I put it in the 2018.
Okay. So this is how auditory information or acoustic information is processed. It comes into the brain through the 8 nerves. nerve, the acoustic nerve. It goes into the brainstem, and in the brainstem, the first synapse here is in the cochlear nucleus. From there, it travels over to the other side to an area we call the superior olivary complex or superior olive. It then goes through a fiber tract that's called the lateral lemniscus into the inferior colliculus, and from the inferior colliculus, which is still in the brainstem, it goes to the thalamus. All sensory information goes through the thalamus, smell, and from the thalamus then to the auditory cortex. And we can measure how sensory information is processed in the brainstem using the auditory brainstem response. And that is something that is relatively easy measured. It requires some electrodes glued here on the forehead and then some here on the back of the mastoid, back of the behind the ear. And when we then play a sound, we see this very specific wave, which is called the auditory brainstem response. It has different peaks, peak 1, 2, 3, 4, 5. And these peaks are created by neural activity along the auditory brainstem. So we know that the first peak reflects activity of the cochlear nerve. The second peak is then here in the dorsal, in the cochlear nucleus. The third peak reflects the action potentials, the neural activity arriving in the superior olive. The fourth peak is the lateral lemniscus. The fifth is the inferior. So we can basically see how an acoustic or an auditory signal travels through the brainstem by recording these auditory brainstem responses. And that has been done in experimental studies. And what we know for kids with autism is that the auditory brainstem responses are slower and they are smaller. So the peaks are slower. There are these delays here, the absolute delays, but also the delays between peaks. They are increased. So the ABR is slower, travels slower through the brainstem, and the peaks are also lower. So there's less amplification in the brainstem while the signal travels up to the cortex. But this seemed to catch up. So these seem to be mostly in children, and at one point during development, the auditory brainstem response seems to catch up. So it looks like children with autism, or at least many of them, have a delayed auditory brainstem maturation. Because during development, also control children, the ABR becomes faster and becomes larger, but much faster than in kids with autism. So they have this delay. Okay. So that is auditory sensory processing in the brainstem. We can also measure it on the cortical level.
Yeah, just terminology. From your perspective, are auditory and acoustic, are they interchangeable?
Okay, so acoustic is everything that has to do with sound. And as soon as it's a signal within the brain, it's auditory signal. So acoustic is a sound wave. You have an acoustic stimulation that then stimulates the auditory pathway. Thank you for that question, because a lot of people do that wrong. And I'm a little bit of okay, so in the cortex, of course, we can measure auditory signals as well. They are called auditory evoked responses or event-related responses, EFPs. There are a lot of different things that can be measured. It's the P50, the N100, the P200. Sometimes they are referred to P1 and P2 and P3. There's mismatch negativity. We can measure brain oscillations. So there are a lot of different things that can be measured on the cortical level. But what, in general, and I just want to define it, what we see with many of these measures is related to what we see in the brainstem. So we see decreased amplitudes and often increased latencies. But this is a very rough simplification. So depending on what kind of potential, actually we published a review last year, which summarizes all these findings, both in humans and in animal models for autism. Okay, so this is auditory processing brainstem, maybe auditory brainstem response, and through EEG we can measure this in the cortex. What about the sensory reactivity? So that is something what we also measure, and Renee, who has been here several times, who could not be here today. She does that in collaboration with Rob, now in children with autism and control children. And the idea is to measure an implicit response to acoustic stimulation. So what we use is an acoustic startle response, which is sudden, short, relatively loud noise. So it's like that's what it sounds. But it's relatively loud. And what happens is that we startle, and every one of you will startle. 90%. I'm a non-startler. I don't know what that means, but I do startle as well, but not to these sounds. But so some people are high startlers, some people are low startler. Whether this has to do with anxiety levels, debatable, or stress levels. But all people do startle. And one component of the startle response, so startle response means, I mean, you all have startled, right? You do that. Close your eyes, your shoulders up, your head, you protect your neck, basically. And what we measure is the eye blink. The eye blink is one part of the startle response, and it's easily measured. It's 2 electrodes here on the muscle, and one sometimes on the forehead, like a reference electrode, and then headphones on. And if we play this sound, this is what the EMG looks like, the electromyogram of this eye muscle here. And then we can integrate and filter it, and we get this nice amplitude startle response. So we can measure how much someone reacts to startle responses. And in general, what we know, not from Renee's measurements, but from the literature, is that a lot of kids with autism show greatly increased startle responses, and often a delayed onset of the startle. So slower, but higher. And that is interesting because because the startle response, so that is an implicit response. You can't help it. You startle. You don't have to learn that. You don't do that voluntarily. This is a reflex. And that is mediated by a very short pathway here. So we have the hair cells in the ear. They are innervated by neurons that then project to the cochlear nucleus, which is here. So in the brainstem, and from there is a connection to the reticular formation here, and there we have motor neurons, or these neurons in the reticular formation directly activate the motor neurons in the spinal cord. It's a very short pathway. It has to be short, right, because it's fast reflex. But it is basically the startle response is a readout of what happens with acoustic information or auditory signals up here in the very lower portion of the auditory processing pathway, namely here in the cochlear nucleus and the cochlear nerve. So it's a readout of, behavioral readout of auditory information in the cochlea. And if you remember, auditory brainstem responses are decreased. The amplitude are decreased in kids with autism. But their startle response is greatly increased. What does that tell you? That, okay, up to here the signals are actually decreased, but then the motor signals increase. So something actually changes in the interface between sensory information and motor output. So it's not only a sensory issue if they started more. It might actually be the reactivity is different. That we could see, but the problem is it never has been measured in the same kid, like both of them. So it never has really been related to each other. We also measure 2 types of sensory filtering. So startle alone tells us a little bit about the reactivity on the brainstem level, brainstem mediated behavior. But we also can look at sensory filtering. And sensory filtering, that is, again, the reduction in sensory signals that take place in early processing pathways, so in the brainstem. And there are 2 things we look at. One is habituation. So if I startle you over and over again, you will probably not stop startling, but you will startle less. First time it's really high, second time it's half of that, third time it's a little lower, and then you stay on a really low level. We call that habituation, and this is what we normally observe, but not in children with autism. So, and some other populations, by the way. So, schizophrenia is another case where people tend to not habituate. they instead even sometimes sensitize to the stimulation, which of course can cause higher startle responses overall because they don't decrease. And the other thing we're looking at is pre-pulse inhibition. That is, if you play a startle pulse, you will have a startle response. However, if you play a pre-pulse, and it could be a visual, an acoustic, vestibular, any kind of touch, any kind of sensory stimulation shortly preceding the startle pulse, and that pre-pulse does not cause startle reaction or anything. It's just a very subtle sensory thing. The startle response will be greatly reduced by presentation with pre-pulse. And this has, again, nothing to do with learning. We are talking milliseconds here, like 100, 200 milliseconds at most. At this reduction in startle, when there's a pre-pulse, we call it pre-pulse inhibition. And that has been developed originally in people with schizophrenia, because actually in the German language, trained in Germany, schizophrenia was actually named filtering disorder, Filterstörung. Filtering disorder. So, and this was like 60, 70 years ago. So people already recognize, psychiatrists recognize that they seem to not be able to filter sensory information. And this is actually called gating. So there seems to be a gating deficit in people with schizophrenia, but we see this in autism as well. So the idea is that normally some unimportant information is gated out. It does not reach higher brain areas. And this gate somehow doesn't work in people with schizophrenia and autism. And so their brains are flooded by sensory information. Again, it's a lack of filter. So we see that in people with autism that there are disruptions of both habituation and pre-pulse inhibition. Okay, so much to the behavior. We have more, oh yeah. So that was brainstem behavior, and the last portion is then cortex. If we look at the cortex, again, we have all these P50, P200, these measurements for the EEG, but of course we can also run perceptual tasks. And someone who did that a lot was Ryan Stevenson, who has been talking here a little earlier, like several weeks ago. So they do all these perceptual tasks, like rate discrimination tasks that looks at frequency, so that's about temporal processing. Again, remember the auditory brainstem response is slowed down. So you would expect them to have some problems with fast timing of auditory signals. So they often get different frequencies, and then they have to distinguish them. And if they're too close together, they can't. They hear like something like, or they hear, and then for them it's difficult to tease that apart. Or a loudness rating task where they have to say whether something is loud or not so loud or very quiet. And kids with autism will consistently rate stimuli as louder as control typically developing children will do. So again, you see this hypersensitivity, at least in some range of acoustic stimulation. And again, Ryan Stevenson, Mark Wallace at Vanderbilt, they did a lot of tests there. So there are all kinds of disruptions in both loudness and temporal processing on the perceptual level in kids with autism and also in adults. Good. So we actually, me being an animal researcher, we started out, and this is from one of the grants I have, a CFREF grant. Our idea was this specific grant focuses on temporal processing, but so we thought we'd take people with kids with autism, and we take a genetic rat model with autism, and what we try to do is we use the same behavioral paradigms. Here it's gap pre-pulse inhibition, which is a temporal thing, but we do that with loudness and other things as well. So we have a behavioral paradigm on the brainstem level. We have a behavioral paradigm on the perceptual level, which relies more on cortical processing, which is here the rate discrimination task, but we could do that with loudness as well. And we run the same in both the humans and the rats in the way that we can actually compare them. And then we take all kinds of different measurements, and this we do mostly, of course, in the rats. We can do all kinds of electrophysiology. We can stick the electrode right into the brain and then measure related signaling. We can inhibit some neurons using Dretzel optogenetics. We can, use pharmacology and inhibit receptors. We can also take the brains out and look afterwards what is different and how do the other pathways different fire. But we can also, of course, look at sensory testing, ABRs and brain waves in the rats, and do some cognitive testing, some higher other tasks, some social behavior stuff, and see how these things are related. And the goal is to parallel as much as we can that's with human findings, but in terms of going into the mechanisms, we are, of course, pretty much restricted what we can do in humans. We can measure these event-related potentials, like auditory brainstem response, ABRs, or we can do EEG, and we can do functional MRI. But that's pretty much what we do. And whether we can do that with kids with autism is difficult. But we can also do, we normally do the IQ testing. We have the psychiatric diagnosis, so we could see how these sensory issues relate to other the psychiatric diagnosis, and see whether this could be a diagnostic marker for certain subgroups. So that is what we try to establish. The big question is, of course, our research is only as good as our rat model. So what model do we use? And so we got the CFRAT funding, and I just got today, I heard that I get this funding.
Good.
So what we use is a catnap knockout rat. So what is the catnap gene?
Should have been called catnip. It would have been much better.
Yeah.
It was a catnip gene.
It is called catnap, but it's not written catnap. It's called contactine, or I don't know. I don't even know what it stands for. It's a long word. It stands for a protein which is called Caspr2, and Caspr2 is a neurexin. So it is a cell adhesion molecule that brings different neural cells together and makes them connect to each other. So as you can imagine, it's very important in development. It's highly expressed during development, specifically in the sensory pathways. And the reason why there is actually a knockout rat, and it was financed by Autism Speaks to create that rat, is because there is a population in Pennsylvania of Danish people where there is where a lot of children, and in one family, I think it's 4 out of 5 children, have a homozygous complete loss of function mutation of this gene. So this is basically a knockout of this gene. This gene does not work in these children. And these children suffer from extreme neurodevelopmental delays and disorder. They become verbal at one point and then they lose it. So they have developmental language disabilities. They have huge cognitive impairment. They have sensory issues. They have social issues, of course. So they have core symptoms of autism, but they're rather of the severe and low functioning end of it. They also get seizures, especially if they grow up. So puberty, late puberty, they get a lot of seizures. Yeah. So it's a severe form. Single nucleotide variants, so other mutations that are not homozygous, not complete loss of function, but single nucleotide variations that might impact the function of catnap2, that might just lower it or increase it or whatever, they are also associated with the risk to develop autism.
Do you know where the genome of that gene is?
It is the biggest gene, and it's in that section. What is that? Where there's a lot of which it has been implicated. Yeah, and it's one of the biggest genes in the human genome. So again, it codes for Caspr2. It's expressed during development, almost all sensory pathways and the cortical areas. This is rare, single gene cause, so it is rare. A double homozygous knockout is really rare. And I mean, it's no coincidence that it appears in Amish people. But again, single nucleotide variants are associated with it as well. So we got this rat. And so we try to do everything that I showed you before, the ABR, the auditory brainstem response, the cortical processing, and all the behaviors in this rat to really then maybe able to look what causes these changes in the rat, and then to see whether we can translate that into the human situation. So we do, and what I will talk today about, so now comes the really scientific part, and I'll try to keep it light. What I will show you so far is the auditory brainstem responses and the startle reactivity and the sensory filtering in this rat. So only the brainstem portion. We are still working on the cortical portion of the perceptual task. So I'll only talk about what happens on the level of the brainstem in these rats during development. So we did, of course, auditory brainstem responses. And again, in children, we would, or in humans, we would put electrodes here and here, and then we get headphones, and then we can measure these auditory brainstem responses in rats. We just stick the electrodes right into the rat. The rat is anesthetized. It can, because all these sensory pathways are not affected, not changing when the rat is anesthetized. And there's a loudspeaker here, and then we play the sound and record the auditory brainstem response. And then we measure the startle response. We do not measure the eye blink in the rat because it would be rather difficult. We can do that, but an easier way is we just put them into these tubes, which are comfortable tubes. They're the same they sleep in their cages, same size. There's a loudspeaker here in the back, and the tube is mounted on a motion-sensitive platform, which pretty much like a balance. When the animal startles, so we play the sound, and then the animal does that, and this moves the platform, and we pick that up with a transducer and computerize it and so on. So we can measure the startle responses. So what do we see in rats? So first of all, first thing that we did is, of course, we measure the auditory brainstem response. That's what it looks like in a rat. It's almost very similar to the human one. Peaks are a little bigger or smaller as compared to the human ones. The brains and the ears are located differently as well. So we measure the auditory brainstem response, and we can start with very low clicks, very quiet ones, and then get louder and louder. And then we see when there starts to be an auditory brainstem response. So this is very quiet at 20 decibel, and then it gets very loud up to 90 decibel, and you see how the amplitude increases. But using this, we can basically determine their threshold. When do they hear? Where's the hearing threshold? And what we find is that, so blue is always the control animals, and red are always the catnap animals. That the catnap animals, no matter if we use a click or a 4 kilohertz or 20 kilohertz tone, so different frequencies, they don't differ. And that is true in very young animals, 28 days, in adolescent animals, 42 days, and in young adults, 70 days. So they are the same. They have same hearing threshold, which is good news. So they don't have a hearing impact. And that's important because catnap is also expressed in hair cells. So that, it could be that they're just dead. Then this would have been for us an absolute no-go. Switch our animal model. No, so they hear fine. The hearing is great, at least on the ear level. Then they have normal hearing thresholds. Then we look at the amplitude of the auditory brainstem. So in red, in blue is the controls, and red is the catnap animals. And as you can see, their amplitude is much lower. And this is not so much true for peak 1, but peak 2. So this is peak 4. So that's this one here for control animals, and this one is for catnap animals. So catnap animal peak 4 is way lower. That means the cochlear nerve and the cochlear nucleus, which is peak 1 and 2, they're still fine. They are not different from between catnap and control animals. But then the higher brainstem areas, like the superior olive, the lateral meniscus, and then the inferior colliculus, these are impacted. So there is less of an amplification of sound in the auditory brainstem. So they have a lower auditory brainstem amplitude, which is exactly what we see in children. And if we look across age groups, this is very pronounced in the very young animal and adolescent animal, but by the age they become young adults, it goes away. So they are fine once they are adult. They look not different from controls. So it's only during development that the auditory brainstem response is reduced.
But adolescents or adulthood, how do these rats compare to the wild type rats in other ways? And so their response to noises is the same by adulthood, but in other ways are they similar to other rats?
They get, like the human situation, by 3 months of age, postnatal day 90, they get so many seizures. You get into trouble with animals.
So their brainstem responses are normalizing, but nothing else.
No, and I will get there. So the brainstem response normalizes. Well, stay tuned. We look at the delay as well, so the speed of transmission, and what we see again is, so this is postnatal day 28. This is phase 1, 2, 3, and 4, controls, and then in red is the catnaps. So they have a longer delay to the peak of the waves. So this is true for wave 1, not so much, but then 2, 3, and 4, and it actually adds up. So the auditory brainstem responses is slower. And if you look at age 42, it's still slower, but not so much anymore. If you look at postnatal day 70 young adults, it catches up. So again, we see slower brain waves, but they catch up by. And we also look at the interpeak latencies between 1 and 2, and 3 and 2 and 4, and we see the same trend. So they're all different at postnatal day 28, but then 24, only this one is different. And at adult, we still have a small difference here, but I would bet it eventually will go away. So we seem to have, like we see in children, this delayed maturation of the brainstem. But we can really now see where exactly in the brainstem problems occur. What about reactivity? So again, I mentioned that in children we have these lower brainstem waves, but then the reactivity is actually increased. So we look at the rats, and this is an I/O function. So we started them with increasingly loud startle stimuli. Starts at 65, they don't startle. 70, they don't startle. 75, they don't startle. 80, they still don't startle. 85, they start. So we see an increasing amplitude of startle responses, and these are young animals, and these are adult animals. And there are males on the top and females on the bottom. This is the only place where we actually see sex differences. So I show males and females separately. But what we can see is that, well, both have maybe a trend in young that they startle a little more, but not so much. But then in the adult, when the brainstem normalizes, the brainstem, the auditory brainstem response normalizes, there we see this huge increase in reactivity. It almost looks like they compensate for the lower brainstem responses during young age, but then the brainstem catches up. And now this compensation creates the problem that they now startle more. And we see this marked left shift of the I/O function. So they show this. Most importantly, they show hyperreactivity to this medium range stimuli, not so greatly much with the really loud ones. It's in here where the huge differences are, right? And a little bit more in males than in females. So that's interesting. So that shows that there is greater reactivity, and that is most pronounced in the lower intensities. And that is exactly opposite of what you would expect if you would say startle is just a readout of the auditory signals in the brainstem. It is something that goes wrong between the sensory and the motor. So the sensory motor connection is somehow different. We look at habituation, just to make it really short. We see normal animals habituate. Catnap animals do not seem to habituate at all in young, but this is something that seems to normalize during the development. That means the lack of habituation does not necessarily cause that higher startle reactivity in the adults. We also look at repulse inhibition. That's a very busy slide. But what the take-home message is, so this is the amount of inhibition for different types of repulses. We have four different types of repulses. We have 75 decibel, the 30 milliseconds before the startle, 75 decibel, 100 milliseconds before startle, and then 85 decibel, 30 milliseconds on. And we do see disruption in all of them in the adult. Here, there's less inhibition, so it's lower in the catnap, the amount of inhibition that is plotted. In the young animals, not so much. We see the trends, but it's not as marked as it is in the adult. And what we also see in the adult, again, this is the baseline startle, the trials where they don't get a repulse, and which goes hand in hand with the IO function I showed earlier. So they have slightly increased startle in young animals, but it's in the adult animals where we have this hugely increased startle response. They also are a little hyperlocomotive, by the way. And then we do social tests and all kinds of other tests with them as well. But here I focus on auditory stuff. So, what does that mean? So, in summary, we were actually amazed how well the catnap 2 rats recapitulate the situation that has been reported in autism. However, the situation or the literature that autism is really, really difficult because people use different paradigms to measure startle, different ways to measure brainstem responses. They have different subpopulations within the autism spectrum. They have different age groups and so on. So it is very hard to actually really connect things. And so this is one reason why we would like to set a standard for both human and the animal measurements, and then see to what extent we can really then translate animal to human research. But what we see in a nutshell is we have normal hearing thresholds in the catnap rats. We have decreased and delayed auditory brainstem responses in youth that then catches up in the adult. We have an impairment of habituation in the adolescent, but not in the adult. But then in the adult, we have this increased reactivity and disrupted repulse inhibition, which is actually more prominent in the adults than during adolescence. And it looks like that these persistent changes might be a result of these early disruptions in brainstem processing, where then the sensory motor structures might compensate for these slower and lower amplitude auditory brainstem processing, and that this maladaptation then, or these compensatory adaptations then cause the problems once the auditory brainstem catches up in maturation, that these maladaptations then cause the problems in the adult, which is a little bit of a hypothesis still, which we of course want to test. So what we want to do going forward is, again, we want to make this connection between animal models and children and adults. With autism, we work with Rob, Brian Stevenson and Jen Sperry as well. Brian Allman, I have to mention him because he is the in vivo electrophysiologist, more the behavioral part. He's more the cortex, I'm more the brainstem person as well. So we together work on this project. So far we have basically validated the catnap2 rat model, that it has sensory processing disruptions, reactivity is altered, sensory filtering is disrupted. We do have quite some cognitive tests with that rat as well that I didn't speak about this, but it shows the same disruptions in terms of loudness and temporal processing, shows stereotypies, it gets seizures later on, it shows disruption in social behavior, and we already looked at the brains, so we started to look at the brains, and we see some of the things that have been reported in post-mortem brains of people with autism, namely that there is reduced GABAergic signaling, and so the ratio between excitatory and inhibitory signaling is different. There's less inhibition, more excitation, which also might play into the fact that they are more prone to get seizures. What we now want to do, of course, is we already started doing electrophysiology in vitro and in vivo. So in vivo, like the ABRs, but we also stick electrodes to the cortex, measure or into different areas in the brainstem, right in there, in the anesthetized, and eventually also the freely behaving rat, and measure signals there, which we can't do obviously in humans. Thereby look at mechanisms and pathways, do all kinds of histology. We want to look at inflammatory markers as well. Obviously, we can do all kinds of pharmacology now, infuse drugs into specific brain areas, or inject them systemically, and or activate or inhibit certain neuronal groups and see how they contribute. And the goal, of course, is to find new targets to intervene, because I think, if anything, our results so far show that the problem seems to be the early development when the sensory processing is disrupted, and this might then cause all kinds of problems later on in terms of behavior that are then not that seem to compensate for the disruption in the brainstem or cortical processing within a window where developmental window where there's for high plasticity. And then as the brainstem catches up, there might not be plastic enough to decompensate or whatever you want to tell. And so these problems manifest. So that would call for early intervention, of course, that you have to do something early on to not even bring the brain into that situation. And so, of course, one thing is we look at drug targets, whatever neurotransmitter we can just antagonize or whatever. But there's also, there are two papers now where they do kind of a sensory enrichment during early development, which probably is the animal version of doing all kinds of occupational therapy and sensory exposure. That shows really amazing results that changes the way the brainstem, not only the reactivity, but also the way the brainstem processes sensory information.
Is that in a specific type of mouse? Rat?
That has been shown in rats and mice.
I think with Fragile X syndrome, I think they've done, with knockout Fragile X, I think they've done something showing.
I think one is Fragile X and the other one, I don't remember which one is, but Mercenich did some stuff there. And they show that this multisensory exposure to, there's gravitation involved, sound, visual stimuli, that this kind of enrichment has tremendous impact on sensory processing, sensory filtering, and reactivity. So these are the things that we want to do in the rats. And I think that's all. Yeah. So I just want to briefly mention Renee, who has been here before, mostly with her daughter, sitting somewhere in the back. So she's the one who measures kids with and without autism or ADHD, where we look at startle, at habituation, and pre-pulse inhibition. And then Kayla, who is a PhD student, co-supervised between myself and Brian Allman, and she does everything I showed about the catnap rats here was basically her work together with some undergraduate students.
If I could just talk briefly about what Renee is doing, because I think it's really elegant. So what Renee is doing, so in the picture that Susanne had with the person sitting there with the headphones on and the electrodes, I think difficulty potentially becomes if people are doing different things, there's going to be different input at that time where you have different visual input, which may have some impact. And so what we're doing is looking at kids with autism, ADHD, or neither, and what they're doing is watching a video during that time. So there's no sound from the video. So they're getting sort of very similar. I guess you can't control for ambient lighting as easily, but they're getting similar visual input and similar auditory input at the same time. And what happens is there's no sound, and the headphones are, I think it's a white noise in the background.
There's a little white noise in the background.
And then periodically there's that sort of click. The difficulty, I think, and this sort of becomes really difficult, is as people get older, you want to try and control for that visual input as much as possible. But having 16-year-olds watch 2 Piedino is going to be a real problem. But I don't know how to really control for that. But I mean, it's funny. I've been impressed with some of the kids who've been able to sit and watch things that I wouldn't have expected.
So she measured a little more than 40 kids so far,
Yep.
and there's some attrition. So there were some kids that just could not.
But surprisingly, I think mostly it was kids with ADHD, which maybe isn't that surprising. But I would have thought with the noise that it'd be the kids with autism who wouldn't tolerate it, and it's actually more, I think,
interesting. Yeah. None of the control kids had problems.
I've got a couple of comments. One, I'm a little skeptical about the use of the catnip kids as a model for autism. They sound more like the kids that have Rett syndrome, childhood disintegrated psychosis. So I think you're seeing more of a brain damage type of phenomenon as opposed to what we see with autism.
But I think that's a really good point, but because they're also, it sounds like they're really, really low functioning. So they're not representative of most people with autism. They're sort of extreme group, perhaps.
No, and we nevertheless chose them because they have these. So because of the language, we actually chose them because of the language, because they have this severe form of language disorder, which is strongly connected to the auditory symptomology. But they do show, so we don't, and normally I don't sell them as an autism model. I sell them as a Rett model that models some of the core symptoms of autism. So this is how I phrase that in grant applications, a bit more cautious, because I mean, I think there is no animal model that replicates autism or schizophrenia. These are intrinsically human conditions to some extent, but we focus on the sensory symptoms, and so we need an animal model that shows the same sensory symptoms, and this is why we chose this model.
Is there
another comment? I looked up an article today on P50s, autism, and Asperger's. Apparently, this particular article didn't show any abnormalities in the autism group, but it did with the Asperger's. And they went on to say Asperger's are mostly males, and therefore there's a question as to the effect of androgen versus estrogen. That perhaps it's the excess of androgen prenatally that might be producing these abnormalities in process.
And the nice thing is we could, of course, now test. And the nice thing is that we can now test things like that in the animal, right? We could influence the hormonal status of an animal and see. And we do, that's also a reason why we test males and females. And I think I mentioned that. So if I say this is disrupted in autism, that means there are reports that certain groups within the autism spectrum have disruptions there. But again, I think we all know that autism is not one disorder; it's a huge spectrum. And so I think it's always a subgroup that shows these specific types of symptoms. And this is what I meant when I say maybe one day we can actually use these kind of things as a diagnostic tool as well. Say, okay, this group has sensory issues, another group might not. But really depending on, what form of autism they have.
The questions?
Yes, I was just going to comment too. It's very interesting because there is a group of kids who are diagnosed with autism who ask parents about that early history. Are these kind of quiet children that require a lot of attention? And if they're the first child, often the parents don't really notice. And so then your treatment, which is this sort of enriched environment, could work. But it's not every child with autism. So my question is, are there other rad models that have autistic-like features and have the same results in terms of reactivity and processing and ABRs found in those as well?
So no one has looked to that extent that we are looking, but I would, of course, so there's a Fragile X rat now, which is another rather severe form. And what we know from the Fragile X mouse, they show very similar things. But now there's a Fragile X rat. So if I get a lot of money one day, I would love to, it's always keeping a rat colony of transgenic rats. So we need to get a grant with that.
So transgenic, not something else.
Yeah, transgenic.
It's a little personal joke for myself, so not transgender rats.
Not transgender, no, transgenic. So yes, but they are all pretty much severe forms, right? So these are, I mean, they are all the genetic, the rare causes where we have a real genetic cause. And there's a handful, like catnip and Fragile X, probably BK channel knockout is a autism one as well. But so there are maybe 4 or 5. And these are all severe causes. We do work with a different model where we do all that as well, and that is poly-IC model. So we work with 2 different models. One is poly-IC, and one is valproic acid. Valproic acid might be easier. That is an injection of valproic acid into a pregnant rat, and all the offspring actually shows weird things. They are socially awkward, and they have cognitive issues. And so there are a well-established autism model. Again, I don't like this word, but the reason is valproic acid is an anti-seizure medication. It's very potent, very inexpensive, and it works for a lot of people where nothing else works. So women take valproic acid even if they are pregnant. There's often no other way to avoid seizures during pregnancy, which are obviously very dangerous as well. But it has been shown that valproic acid increases the odds that the child will develop autism 7-fold or something like that. So that is one that led to this valproic acid model. And then the other is poly-IC, which is a synthetic molecule that does not do anything. It's totally benign. But for our immune system, it looks like a bacteria. No, like a virus. Sorry. No, like a virus, because it looks like a double-stranded RNA. So our immune system, or the rat's immune system, thinks that there's a viral infection. It ramps up an innate immune response. And it has been known, and there are a lot of studies showing that viral or bacterial infection during early pregnancy, first trimester, second trimester, at the beginning, increases the risk of the child to be autistic by, I don't know how much, 3-4 times or so. And that led to this model where poly-IC is injected, the innate immune response is ramped up, and that causes damage to the brain during that developmental stage. And so both of these models we have in my lab as well. And we look at all of these, not all, but a lot of these in these rats as well. They are not as severe as the catnip. So it's a little, and it's hard if you have a mild phenotype. You want to have a strong phenotype for these kind of. Yes, so if they either don't show that or they show the same. So some of the things they don't show are very subtly only, and then some of the other things I showed, they show as well.
But it goes to specificity though, because I think so when you talk about women who have viral infections at a certain point in pregnancy have increased rates of autism, I think it probably goes to schizophrenia as well. Mm-hmm. And we know that malnutrition at certain points of pregnancy can cause later rates of increased schizophrenia and I think autism. And so let's figure out what the specificity is. And so with those catnip mice, you said that humans can be heterozygous.
Yeah.
Are there any heterozygous mouse models that wouldn't be as severe?
So we measure the heterozygous as well.
Yeah.
That's by the way, not mice.
Sorry. That's the same thing. I don't like.
No, it's not the same thing.
I don't like either of them in my house.
I want to make it really clear that a mouse is not a small rat, vice versa. They are super different animals. Like cognitively, they are so different. They are as far apart from each other as the rat is from the human. The mouse is double the distance from the human as the rat, genetically and behaviorally and cognitively. So it is really a different animal.
So with the rats then, the heterozygous rats, how do they look?
So we have the heterozygous, and they are in many cases look like wild types. They have some small changes as well, sometimes opposite, which kind of throws us off. But yes, we do.
But in humans, the heterozygous people have an increased risk of autism.
I'm not sure if they have enough to actually say that, because the parents of these kids are both heterozygous carrier, right? Yep. And they seem to be fine. So at least not severely diagnosed. Yeah.
So our OT is working in this area with sensory processing, and we think about 7 senses versus 5. Okay. So the proprioceptive and vestibular. Mm-hmm. And with children with autism, we see atypical reactions especially the movement sensory system, where the children with autism tend to have a very high threshold for movement. They can tolerate a massive amount of movement with little reactivity as well. So we do a measure of post-rotary nystagmus, it's called, looking at the back and forth eye movements following spinning.
That's interesting because we know the brain circuit for nystagmus, right? So that would actually be really interesting to look at as well.
Is that why some kids we see can spin and don't get dizzy?
Spin and spin. And then that also tends to relate to poor motor development because they'll have poor vestibular discrimination skills too, right? And then together with that proprioceptive system or the body awareness system, the receptors in the muscles and joints, both of those also lead to very poor integration of those inputs, leading to poor feedback then from the body, leading to poor motor input.
That's very interesting. So that's why I hang out here sometimes. It's actually to learn. You know, I work with rats, so I always talk about clinical stuff.
So we have a big sensory motor gym that would be a rat version of the great cage, right?
Yeah.
Because I talked with Christine Hodges. She was a figure skater when she was young, right? And I've been trying to figure out why can some of our kids, because I don't think they spot when they spin. So it's not clear why they don't get dizzy, and I guess that makes sense. So you've answered that question. It's nice to have one question definitively answered.
Yeah, so you can see it in kids without autism as well. So the children that I'm working with on Unit 5, a lot of them will have very limited post-motor.
But I guess they're not typically developing kids, regardless. So it's not.
Yeah, but there's differences in the sensory processing pathways for having that brain.
So when Susanne was saying earlier,
And the nice thing is that just the nystagmus. I mean, this is the inferior colliculus here is heavily involved. The inferior and superior colliculus.
So very interested in the vestibular system. And we see it in sensory processing and sensory integration as sort of the overarching sensory system. And when we make impacts with that sensory system, we see changes in lots of other sensory.
There's of course very little animal research about vestibular system because it's just not as easy to explore and as acoustic to just play sounds. I don't know, spin a rat. We would probably get into trouble, which is stupid. Why wouldn't we spin a rat? But, and then the rats probably don't have nystagmus because they don't have. They might or might not.
So interestingly,
One question
always leads to
it's an empirical question. Yeah.
Put an undergraduate student on it.
in terms of thinking about treatment then for children, like in California, if little small children have at risk for autism, they go into a kind of specialized preschool program there, an early intervention preschool program, where they have physio and speech language pathology and OT all directly in it every single day. And they're usually set up right within the therapy clinics there. So I think a lot of those children make huge gains.
But I think the difficulty with at-risk kids is you don't know for sure which those kids would have
But they're at risk for
autism.
something. They're at risk for developmental delays.
Okay, so they get a show growth.
Yeah, like
And they show that
at-risk
compared to
for
kids that
delays.
aren't involved, they show improvements.
Yeah, and it's an integrated preschool program too. So typically they'll.
Because I think they've shown that with mice or rats, one or the other with Fragile X, rats.
I think so, yeah.
But I think it's easier because we know exactly what the neuronal abnormalities are. And I know I don't know if that's been shown with children with Fragile X syndrome. Would you know, Claire? Because I think with rats with Fragile X, with knockout Fragile X rats, they've shown that a significantly enriched environment, there's a significant improvement in development. And I don't know if anything's been shown like that in human beings. So it's obviously much harder to control.
Yeah, I would not, be too much fixated on specific genes because what all these genes do that cause autism or the innate immune response, they impact the same developmental processes. It's all about the wiring of neurons, the synaptic stability, and sometimes also the wiring of myelin sheath, which.
But I guess it hasn't been shown. Has those kind of things been shown to improve with kids who have those things? Do you know anything?
So if you put Fragile X kids in an enriched environment, do they do
Yeah,
better?
so I think you put it like what I'm thinking as you're talking. I mean, in really old studies of rats show we put the rats in an enriched environment, and they have more neurons and connections. So there's no reason to think that anyone with these developmental risks would not.
Yeah.
So it's much harder to control
kids
in
in enriched environments do better than kids in non-enriched, and kids at risk any of the studies.
Hydraulics is not-
These are fragile X tend to have the really numerous areas of increased hypertension.
But there's so much more. It all
Yeah.
has to be.
Yeah.
So my last question is: these studies been done in rat models of alcohol?
No.
Because they have some similar later reactivity, but clinically they have earlier, quicker reactivity.
Okay. So that's fetal alcohol syndrome, or not that I'm aware of. I don't look particularly for this.
But it is one of the things, like everybody has their model when they do these stuff.
Yeah.
Their model they have different sets of tests.
And I mean, you could argue that fetal alcohol syndrome is a similar neurodevelopmental.
It would be a model of a diffuse brain versus.
And in practical, it's just my own experience. They make less things than autistic kids and other kids.
The problem is probably that this is more like a chronic lesion, as opposed to like even an infection during pregnancy is like the innate immune response lasts 2 days or 3 days. It's not the pathogen, right? So unlike Zika, which impacts the brain, the virus infection or bacterial infection, it is the mother's immune response that does the impact. And we actually have a small project with a guy who does placenta research, looking at how actually this impact happens in the uterus. But it's only like a 2-day impact, whereas fetal alcohol syndrome probably is more severe because it's a chronic impact. Right? It's not just one time.
And that's kind of very
So I think of fetal alcohol syndrome as having a motor gating disorder, impulsiveness. So there is such thing as motor gating, typical Tourette syndrome. There's a motor gating phenomenon. More of an impulsive, hyperactive, aggressive.
But the person who I emailed you about, Emma Durden, who's going to be in, so the postdoc she wants to hire to doing research with zebrafish and alcohol exposure. So I'm not sure why zebrafish.
Neither am I, but it's big in FASD.
Really?
Why zebrafish?
And who thought about giving fish alcohol also?
But it's easy. You just put it into the tank. That's
Why zebrafish are not
must
goldfish?
multiply faster.
the zebrafish has been fully sequenced, so there are not.
Oh.
So it is one of the genetic models.
So I think clinically, my own sense is at times, I think with the hearing stuff or the sensitiveness, I think some people do improve as they get older anyway. Okay, so the two physicians are saying yes. The occupational therapists are looking at me like I'm out of my mind.
For the auditory reaction?
I think so. I don't think we see a lot of 17, 18-year-olds wearing headphones.
I think we still do.
Clinically, in my office, I don't see too many. Now maybe their parents take them off, or maybe I'm not aware of it. But it just when you're saying that things may normalize as people get older. I mean, I think that's something that. But again, that's an empirical question. We can actually watch.
And is it just in the sensory system?
But also, I think that
It's
it's just a kind of bias because you continue to see the people who are continuing to have problems. And is that why they're continuing to see occupational therapists? I think that's difficult to but it's also because when talking about the delayed but the startle is increased. And I often wonder that because in kids, we ask parents, what's your child sensitive to vacuums or hair dryer? And those are things that often start. And so I don't know how you can distinguish the startle from the
ongoing.
The noise. I'm not even sure in a research paradigm how you'd even assess that.
Well, and this would maybe play into that lack of habituation because this ongoing noise is something that normal people would habituate to, and the question is, do they habituate to that? Some might be able to, and some might not.
I think so those noises are intermittent, though. We don't have, well, some of us don't have hair dryers on all the time.
No?
But it's a difficult thing to assess. And knowing what, because I think the things we talk about are often more startle. So people think, yeah, a fire truck noise will bother my son, or car backfiring, but those are going to bother anybody.
Yeah.
So I don't know how you separate those two things.
Yeah, it's difficult to separate these things. I do agree. I think, so what I would say from our results is that So receptor desensitization or adaptation might not be different because we don't really see any indication that anything is different at the periphery. Sensory receptors, the nerves. The problem really seems to start in the brain. That's one thing I can say for sure. Whether this is a problem of, so I would say habituation normally happens if we have intermittent stimuli, but a constant noise should also cause habituation. And so they might not be able to habituate. But it's actually a good question. We can't measure it because, well, it will cause habituation, like neuronal habituation for sure, because if you play a constant noise, what most of auditory neurons do is they start firing a lot at the beginning and then they calm down. And so that's something we could test in electrophysiology to see whether there's a difference. Something we don't really get the way we test stuff.
It'd be interesting if they habituate to that background sound, like the sound of that fan on that computer, right? They habituate to that, but then don't respond as well to a new auditory.
Yeah, and habituation
like
is always specific for the type of sound. So even if they habituate to the background noise, it does not mean that they habituate to the startle. So they are different. You can habituate to one stimulus.
I wonder if it's not even the startle though. Like I would think, like just again with the child, if we have background noise or the fan coming on or the heat coming on or air conditioning coming on, if they don't habituate to that and then don't respond to verbal information.
Well, it might also be a learning thing. And so we see several kids who, if they see children crying or if they hear kids crying or babies crying, they freak out, they go bananas. And yet you think that you learn over time. If I see a kid in a stroller, the likelihood of me hearing a crying baby is increased just statistically. And yet it doesn't seem to make a difference. The fact that people are exposed to that setting, but the noise still bothers them. And so they're not learning the way we would expect people to.
But that's exactly the point. So this habituation does not only occur in terms of startle, it occurs in, of course, all processing pathways, at least that's what we claim. And it is influenced by stress, it's influenced by anxiety levels, but it's also influenced by meaningfulness of the stimulus. So if